All right, so last time, as you'll recall, we left off talking about actin and myosin, the two contractile proteins inside of a muscle cell. So one of the things, or one of the topics that students often do their pathology papers on for this class is muscular dystrophy. So for those of you that don't get the pleasure of reading those papers, I thought I would discuss what dystrophin is and then what happens in muscular dystrophy. So dystrophin is a protein, as you can see there, that links actin to the cell's membrane. So there's some terminology on here that hopefully you'll remember from last time. So remember again, actin, this little blue thing down here, that's one of our two contractile filaments, it and myosin. And then the sarcolemma, that's the muscle cell's cell membrane. And then the basal lamina is a basement membrane that surrounds the muscle cell's membrane. And then not pictured, but up here, we would have then the endomysium. And so effectively what the protein dystrophin does is it connects the actin filament to the cell's membrane and also to that connective tissue that surrounds the cell. So as you can see on my slide, it links everything to the endomysium, to that layer of connective tissue around the cell. And so under normal circumstances, what happens is when actin gets pulled toward the midline by myosin, then that's going to take up the slack here in dystrophin. And so it's going to pull dystrophin this way. And then that's going to cause the sarcolemma, the cell's membrane, to sort of collapse in on itself, because that's going to need to happen during contraction. The cell's membrane is going to have to close down a little bit. And so under normal circumstances, that all proceeds regularly. When actin stops being attached to myosin and then we stretch the muscle back out, then the actin filament moves back toward the right here, and then we put a little slack back in dystrophin, and everything goes back to its normal resting length. Well, if you've got muscular dystrophy, what happens there is that you've got some sort of a mutation in the dystrophin gene. So genes are effectively instructions for building proteins. And so if you have a mutation in it, what that means is you've got a change in the letter sequence. You have effectively an alteration in the instructions. And so if the instructions aren't what they should be, then you're not going to be able to assemble the protein like the body should. So for example, think of it if you got some IKEA furniture and it was missing a couple pages in the instruction manual. What you eventually assembled may not look like the bookcase or whatever it is that you bought, may not look like what it's supposed to. And so it's the same kind of thing here. If you have an alteration in the gene, in the instructions, then you get a differently shaped protein than we intended to build. And if a protein is shaped differently than it's supposed to be, then that changes its function. So in the case of muscular dystrophy, that gene alteration makes for a dystrophin protein that is particularly fragile. And so effectively, dystrophin, one of the ways to think about it, and I'm not going to get into any of the detail on any of this stuff, but all of this is the dystrophin protein. And so effectively, if that protein is faulty, it can't act as a shock absorber for muscle contraction. So effectively then the actin filament is going to pull on dystrophin, and maybe what ends up happening here is that some of this part of the protein pulls out of the cell's membrane. And so you can see there the wording integral proteins. Integral proteins are proteins that are embedded in the cell's membrane that have to be there. If something is integral, you can't do without it, right? So integral proteins are proteins that can't be done without. And so if dystrophin is faulty and pulls away from the cell's membrane, then that's going to cause pretty significant damage to the cell. That's going to cause inflammation. And so what ends up happening then is you get some cells like neutrophils, so your white blood cells effectively, that are going to come to the area and they're going to try to clean up that damage to the cell. Well, that's going to happen essentially every time the muscle contracts. You're going to get this profuse damage throughout the muscle. And so if you keep damaging your muscle cells every time you contract them, then eventually those muscle cells are going to die. And so to show you kind of what that looks like on a cellular level, so that's a normal muscle there on the left. And so obviously all of these are your little muscle cells. And you'll notice how different the picture is over here on the right. So you've got a normal muscle cell here, but these white things are fat droplets. And then all of this stuff is connective tissue. All of this stuff is collagen. And then these little purple things here that are all clustered, those are all a type of immune cell. So they're in the area kind of cleaning up the damage. And so with that repetitive damage, again, to those muscle cells, eventually your muscle cells are going to die, and they're going to get replaced by connective tissue and by fat. And so if you infiltrate a muscle with fat and connective tissue, it's not going to be nearly as strong. And so this particular, the most common type of muscular dystrophy is called Duchenne muscular dystrophy. And so that particular one, Duchenne is the one pictured there in the top left. That's its presentation pattern. But it is a sex-linked condition. And so what ends up happening there is, so think back to your biology class. So you've got two sets of chromosomes, your X's or your Y's. So males have XY, and women are XX. And so this, for Duchenne muscular dystrophy, the mutation is sex-linked recessive. So if you only have one copy of it, then that's going to be problematic, because that's the only instructions the body has or the muscle cells have for making that protein. So this condition is present almost exclusively in males, because males are XY. And so since this is on the X chromosome, if boys or males only have one copy and the one copy they have is wrong, then they can't make the protein correctly. In order for women to develop Duchenne muscular dystrophy, they have to get 2 copies of the mutated gene, which is fairly unlikely. And so because of that, it's much less common in women than it is in men. If somebody has this, so it's typically diagnosed pretty early, between the ages of 3 and 5, and effectively what happens is, as the child is getting older, they become increasingly clumsy. So they learn how to walk somewhere around the age of a year, but they start to be more and more clumsy. They fall over, they lose strength, and one of the other physical signs you tend to see is that they end up with really big calf muscles, is a common presentation of it. And so typically in the case of Duchenne muscular dystrophy anyway, it's going to progress from the extremities upward, and then eventually it moves from distal to proximal. And then as it then gets into the trunk, it'll start to affect the head, chest, and cardiac muscles. And so a common presentation is that patients are wheelchair bound by the age of 12, and many of them die of either respiratory failure or cardiomyopathy by their 20s or early 30s. But Duchenne muscular dystrophy, although it's I believe the most common type of muscular dystrophy, there are other types, and so the other types are essentially different mutations in the dystrophin gene. So you can either add letters to the gene, so you can add to the instructions, and that causes a different shaped protein, which causes a different dysfunction, or you can subtract letters, and so then again causes a different shaped protein and a different set of dysfunctions. And so these other things, B, C on down, are different types of mutations or different alterations in the gene, again the protein building instructions, and so then they present differently. All right, so there's my little bit about muscular dystrophy. So let's talk about muscle fiber contraction. So in terms of what we're looking at here, so I'm going to describe to you the process of getting the signal to the muscle cell from the central nervous system that's going to initiate muscle contraction. So the way it's laid out, and this is straight from the textbook, this is not from any of the other sources I've been using, and again this is chapter 9. So the first thing on there is nerve stimulation. So the muscle cell has to receive a signal from what's called an alpha motor neuron. So alpha motor neurons are nerve cells that interact with our skeletal muscle cells and cause them to contract. We'll talk about other types of motor neurons later, but it's important to know that an alpha motor neuron is the type of motor neuron that's going to interact with or attach directly to a skeletal muscle cell and cause it to contract. So the muscle cell has to get input from the alpha motor neuron, and then we're going to get an action potential. So a few terms here. One of the terms we'll talk about, or that I'll use some, is depolarization. So if something is polarized, there are different charges at different ends. So it's positive on one side and negative on the other. So a muscle cell at rest is polarized, meaning that the inside of the muscle cell has a negative charge relative to the outside of the cell. So at rest, a muscle cell's charge is somewhere between -70 and -90 millivolts. So the inside, again, is negative relative to the outside. And so what causes that change in charge is the concentration of ions. So ions are electrically charged particles, atoms in this case because they're singular. So things like sodium, so sodium's Na+, calcium, also a positive, chloride is a negative. Didn't talk about potassium yet. Potassium is another positive. So you've got these different ions that have either positive or negative charges, and so their difference in distribution inside versus outside of the cell is what causes that negative charge. Now, one of the things that is confusing, or at least was confusing for me the first time I learned all of this stuff, is the fact that two of the major players, especially when we're talking about the function of nerve cells, are both positive ions. So in the case of neurons, the primary players are sodium, which is positive, it's Na+, and potassium, which is also positive, it's K+. So the thing that was confusing for me is like, okay, at rest, inside of a neuron, there's some potassium, positive ion, and then there's some sodium, or actually a lot of sodium, outside of the cell. That's a positive ion. And so where does the negative come in here? And so where the negative comes in is that there's so much more sodium outside of the cell than there is potassium inside of the cell that the inside is negative relative to the outside because of that difference in concentration. Way more positive ions outside of the cell, way fewer inside, and so that's what accounts for that, that polarity, that difference in charge. All right, so an action potential then, circling back to the slide, is a wave of change in charge in a cell. So the way that neurons send their electrical signals, because that's how they communicate, is the cell changes charge. It goes from being negative on the inside to positive very temporarily, and then back to negative. And so that wave of negative to positive, that wave of depolarization, or that wave of change in charge, is how that cell then sends an electrical signal. And the same, or yeah, we'll stick with that. The same thing effectively happens in a muscle cell, that the muscle cell is also going to be negative relative to the outside, and then it's going to experience a change in charge. It's going to go from negative to positive very temporarily, and that charge is going to be a wave of depolarization, or wave of change in charge, and that wave is referred to as an action potential. So that action potential has to spread along the sarcolemma. Again, that's the muscle cell's membrane. And then that change in charge is going to cause the release of calcium from the sarcoplasmic reticulum, which we mentioned last time. That's the important thing I want you to know about the sarcoplasmic reticulum, is that it stores calcium. All right, so depicted similarly here. So the way this textbook lays it out, two phases. So the action potential arrives at the neuromuscular junction. So the neuromuscular junction, as you'd expect based on the name, it's the interaction of the nervous system and the muscle cell. So that's the neuromuscular junction. Then the other things on here, so ACH is acetylcholine. So acetylcholine is the neurotransmitter that initiates muscle contraction. So the interaction between the neuron, again the alpha motor neuron, and the muscle cell, there's a little gap between those two. So again, that's the neuromuscular junction. So there's a little gap between those two, so the electrical charge can't spread directly from the neuron to the muscle cell. It has to have something to bridge that gap, and so that's a neurotransmitter, and in this case the neurotransmitter is acetylcholine. So acetylcholine is going to be released. It's going to bind to receptors on the sarcolemma, again the muscle cell's membrane, which is going to cause a change in permeability of the sarcolemma. So the muscle cell's membrane will allow ions in that normally it won't, so specifically sodium, and potassium goes the other way. We'll talk about that in a second. That's going to cause that depolarization, or that action potential, which then spreads along the sarcolemma. The action potential then runs the entire sarcolemma, goes down inside the T tubules, and again causes the release of calcium from the sarcoplasmic reticulum. Calcium then binds to troponin C, moves tropomyosin out of the way, and then actin and myosin, or myosin attaches to actin is probably the best way to say that. All right, so went through that super fast, but I'm actually going to break that down a little bit, and we'll go through it step by step. So a few other things to be aware of. So this is kind of an esoteric depiction here. So what is this? What are you looking at here? So obviously this is a muscle, right? And so then we're looking at a fascicle, throwing it back to last time, so a bundle of muscle cells. And then here you have your individual little muscle cells. All right. And so what is this thing? This is your spinal cord. I mean, obviously everything's labeled, but just to help you out here. So this is the spinal cord. The spinal cord, remember, is the bundle of nerves that relays signals to and from the brain. So in the anterior horns of the spinal cord, which so you can see that the middle part of the spinal cord kind of looks like an H. So this is always referred to as gray matter, and we'll talk about what that is later. But anyway, in these anterior horns there, or the ventral horns, you've got the cell bodies. So this is the main part of one of your alpha motor neurons, and then it has a really long axon, so think of it as an electrical wire, that goes out and attaches to this muscle cell and this muscle cell. So then when this alpha motor neuron generates an action potential, when it sends an electrical signal, that's going to go from your spinal cord all the way out, down here, and then it's going to stimulate this muscle cell, sorry, not muscle, but this muscle cell, and then also this muscle cell. If I stimulate this red one, then it's going to send an electrical signal all the way down its axon, and it's going to stimulate this muscle cell, and then this one, and then this one. So this particular alpha motor neuron stimulates 3 muscle fibers, and the purple one only stimulates 2. And if I activate both of those, I'm going to stimulate 5 muscle cells. So that's what we're talking about. This next picture here, all right, so this is the neuromuscular junction. So I'm going to back up. So what you're looking at on this picture is essentially a close-up of just one of these little things right here where my mouse is, and its interaction with the sarcolemma. So, and you can kind of see that here. So this is the end of the alpha motor neuron, and you can see the little terminals here that are going to interact with the muscle cell. And so now we're close up on just one little axon terminal. So the terminal is the end, the axon terminal, and then so all of this pink stuff down here, this is the muscle cell. All right, so we're going to get this wave and change of charge where the motor neuron goes from being positive to negative very briefly. That change in charge opens up these calcium channels because, again, Ca2+ is calcium. So a few other things, and we'll talk about this in more detail when we get to the nervous system, but there are 2 types of channels that are going to be important for us right now. Those are called voltage-gated channels and then chemically gated channels. So voltage-gated channels are, think of them as a closed door that wall off the inside of the cell from the outside of the cell, and by saying that they're voltage gated, what that means is that a change in charge of the cell's membrane is going to cause that door to open. So as this neuron goes from being negative to positive, that change in charge, where it goes negative through here, when that negative charge hits this calcium channel, that's going to cause it to open because it's a voltage gated channel. So that change in charge causes an opening of this door, and because there's a lot of calcium outside of the cell, out here, calcium wants to move with its concentration gradient. It doesn't like being in a crowded area. It needs its space, as do sodium, as do potassium. So all of these ions are going to move with their concentration gradient. They're going to see, hey, there's a lot of other ions around here that are just like me, a lot of other calciums. I want to get away from them when I get the chance. And so when that door opens up, in this case, again, a voltage-gated channel, calcium is going to go rushing inside. So that influx of calcium inside of the terminal of the axon then is going to cause these little guys, these little circles that have green things in them. So the green stuff is your acetylcholine, again, that neurotransmitter. So the influx of calcium is going to cause these vesicles, which are kind of like little storage bubbles, is going to cause these vesicles of acetylcholine to migrate to the end of the terminal. And once they fuse with the end of the terminal, they're going to dump their contents out. So they're going to dump their acetylcholine into this space between the end of the alpha motor neuron, again the terminal, and then the muscle cell. And so acetylcholine migrates across this space, which is called the synaptic cleft. So it migrates across the space, and then it's going to find the receptors that it likes to bind to, and so acetylcholine will attach here to these channels. And so those are the other types of channels that I mentioned. Those are your chemically gated channels. So a chemically gated channel then opens in response to receiving a chemical. So in this case, the chemical is acetylcholine. So when acetylcholine binds to this little chemically gated channel, it's going to open up. This chemically gated channel is a sodium channel. So not only do you have a bunch of calcium out here outside of both of these cells, but you also have a bunch of sodium. And so when we open these chemically gated channels here, that's going to allow sodium to come rushing inside. So sodium's going to come running in. And so as that happens, remember that sodium is a positive ion. And so if we allow a bunch of positive ions, if we allow a bunch of sodium to move in here, then this area of the membrane is going to become more positive. So once we hit a certain threshold voltage, then, which is somewhere for most cells, -50, -55. So again, if the resting charge of a muscle cell is somewhere between -70 and -90 millivolts, as we allow more and more sodium to come in, because again, it's going to be positive, it'll move from -70 to -65. And then as more sodium comes rushing inside, -60. More sodium comes rushing inside, -55. And then once we reach that threshold voltage, again, probably somewhere around -55 to -50, once the inside of the cell becomes positive enough in this area, it'll open up some other channels right here, which are not pictured on this drawing, but I'll show you a different diagram here in a second. And so there will be voltage-gated channels, voltage-gated sodium channels, adjacent to these chemically gated sodium channels. And so once we get enough of a change in charge as a result of these chemically gated channels opening, then the voltage gated channels open, and so then we get just a bunch of sodium rushing inside, and then that spreads down the length of the sarcolemma. So now it's diffusing, and so to move up here, it's going to diffuse across the muscle cell this way. So the membrane's going to go from being negative to positive, and you get a wave of change in charge this way, and then a wave of change in charge that way. And so that's what all of these letters say without me reading them, but this is a really important little diagram. And so there's our little sodium and potassium. So potassium is actually going to go the other way. And actually, I'll show you, we'll talk about that more in a second on a different drawing. And then what ends up happening, so you have an enzyme out here in the synaptic cleft called acetylcholine esterase. An ACE is an enzyme that breaks things down. And so acetylcholine esterase breaks down acetylcholine. So it'll break down these little green molecules here that are attached to the chemically gated channels. And then once it breaks them down, the channels close. And so no more sodium can come in this door unless we get more acetylcholine. So acetylcholine esterase degrades acetylcholine, and then we close this door. So now no more sodium can come rushing inside. And so if we want the muscle cell to continue to send electrical signals, we actually have to pump the sodium back out, and then we'll pump potassium back in. Oh, and so these are on the slideshow that's up. You can run these as videos, but I left them embedded if you want to watch them. So I won't make you do that now, but in case you wanted to, they're embedded in the slideshow that's posted if you want to watch all of that happen on a video. All right, so we talked about all of that stuff. So the end plate potential is that negative charge of the inside of the muscle cell. Depolarization is where it goes from negative to positive, and then repolarization is where we reset everything. So we go back to that original resting state, that negative charge. All right, so this is what I was talking about a second ago. So we receive acetylcholine, open a chemically gated channel, sodium comes rushing inside. Once we hit that threshold voltage, then these voltage gated channels are going to open. And so they'll allow sodium to come rushing inside. Again, all of these are going to go with their concentration gradient. So they go away from where there's a lot of that particular ion to an area where there's less of it. So there's a lot of sodium outside, so it wants to go inside. Potassium goes the other way. So potassium, there's a lot of it inside, and so it goes rushing outside. And so what ends up happening, sodium comes rushing inside of the cell goes positive. And then what we'll do to try to reset that charge is you'll get the opening of a voltage-gated potassium channel. So you let potassium go back outside, and if we let enough of those positive ions leak out, then that resets the charge of the membrane. The inside of the cell is negative again, but we still have to reset those ion positioning, so we're actually going to pump potassium back in and pump sodium back out. But you can see here, so this is that wave of positive charge, and then here's negative where the membrane hasn't depolarized yet, and then down here we're ready to go again, kind of, where we've got it, the charge at least has been reset, so we're repolarized, and so the inside of the membrane is negative again. So here's another depiction of what happens, just in terms of change of charge. So this textbook uses -90 as its number for the resting membrane potential of a muscle cell. Again, anywhere between -70 and -90. So we open those chemically gated channels, sodium comes rushing inside, then we open the voltage gated channels, and then we get a big change in charge where the cell goes from being, again, somewhere between -70 and -90 up to +30. And then we're going to close those sodium channels, and then we're going to open up those potassium channels, again, in an attempt to reset that negative charge of the membrane. So potassium goes rushing outside, and so then that loss of potassium resets that negative charge, and so then we end up back down here to -90. We close those potassium channels. We don't let any more of it leak out. But again, we're at that initial charge, but we still have displacement of the ions. So then we're going to run our sodium-potassium pumps. We'll pump sodium back out. We'll pump potassium back in and try to maintain that resting charge, but also that positioning of those ions. Again, primarily sodium outside, primarily potassium inside. All right. So then that change in charge, again, that action potential or wave of depolarization spreads along the length of the muscle cell. So it goes this way, and it goes this way along the sarcolemma. And then it moves down the T-tubules, because remember the way I described the T-tubules to you last time was that they are effectively continuations of or extensions of the sarcolemma of the cell's membrane. And so we spread that wave of depolarization both along the sarcolemma and then inside of the cell via the T-tubules. That change in charge then spreads along the sarcoplasmic reticulum. And so once the sarcoplasmic reticulum goes from negative to positive, that change in charge is going to open up calcium channels. And so the sarcoplasmic reticulum then is going to release calcium. And so once we release calcium then, so what you're looking at here, so obviously this is actin, the blue stuff, and this is myosin. Remember, we talked about a couple of additional proteins last time on actin. So there's the actin that myosin attaches to, but the other two proteins we talked about were tropomyosin and troponin C. And so tropomyosin at rest actually sits on top of or obstructs the active sites or the binding sites on actin that myosin has a high affinity for. So when tropomyosin is sitting on those sites where myosin will bind, well, it can't attach to actin, right? There's something physically obstructing it or physically blocking it from doing so. So the other protein is troponin C. And again, I emphasize the C because C for calcium. So when we release the calcium from the sarcoplasmic reticulum, it's going to go down in here, and you can see here the little Ca plus, or 2 plus. It's going to go down in here, it's going to bind to troponin C. When calcium binds to troponin C, it's going to change shape. And troponin C is attached to tropomyosin. So when troponin C changes shape, it'll pull the tropomyosin off of those binding sites on actin that myosin has a really high affinity for. So as soon as myosin sees those binding sites, it'll jump up and attach and pull actin toward the midline. All right, so that's your cross-bridge cycling, your excitation contraction coupling. So the release of calcium, again, it's going to attach to troponin C, changes shape, pulls tropomyosin off of the binding sites, myosin will jump up and attach, pull actin toward the midline. All right, so even more detail. Let's talk about some other things that happen. So what I just described to you was cross-bridge formation. So that's the interaction of myosin and actin. So whenever myosin sees those binding sites or those active sites on actin, different textbooks call it different things, it'll jump up and attach and pull it toward the midline, right? So cross-bridge formation, myosin jumps up and binds to those active or binding sites on actin. And then the next thing that happens is that myosin is going to pull actin toward the midline. That's the working or power stroke, and so that's what's depicted over here. So the first thing is myosin jumps up and attaches to actin, and then myosin changes shape. It pulls actin toward the midline, right? So toward that M line when we're thinking about the structures within a sarcomere. So myosin then is going to pull actin toward the midline, which is what's happening here in picture number 2. The other thing that I haven't mentioned yet this time that we talked about a little bit last time was the energy molecule ATP. Remember, that stands for adenosine triphosphate. And so what we do, so if you have an adenosine backbone and then 3 little phosphate, let's go with, not ions there, but 3 little phosphates, let's just stick with that. 3 phosphates bound to that adenosine backbone. And so there are high energy bonds between each of those phosphates. And so what we do is we break off the most distal, the farthest out phosphate, and then when we break that bond, that gives off a lot of free energy. We talked about that last time within the context of when we give off that free energy, some of that drives muscle contraction, but the remainder of that is going to be given off as heat. And so we talked about the role of muscles in maintaining our core temperature. So after myosin has pulled actin toward the midline, we have to attach a new ATP molecule to get myosin to detach from actin. And then we're going to split that ATP. We're going to break that bond that I just talked about. We're going to give off that free energy. And so that energy that's given off when we break down that last phosphate bond, that's ATP hydrolysis. We use an enzyme called myosin ATPase, which be aware that it's on the myosin heads. We'll probably talk about it in more detail later. But anyway, we use an enzyme called myosin ATPase. We split off that last phosphate, and so the energy given off when we do that allows us to reset the myosin head. So one of the ways I like to think about this is the myosin head at rest is like a cocked spring, right? So it's a mashed down spring. It's ready to go. And so as soon as it has the opportunity, it's going to expand. It's going to jump, however you want to describe a spring, lengthening, if you will, right? So in order to get the spring back compressed again, that's going to take energy. I have to physically mash it back down into shape. And so that's kind of what's happening here with ATP, is that we're going to use some energy, we're going to break down ATP, and then we will reset this spring, which is the myosin head. So the way this is referred to here, or in this textbook, is as cocking of the myosin head. So the steps then, once those active sites on actin are exposed, myosin will jump up and attach to those sites, and that's your cross-bridge formation. And because myosin, or a myosin head is kind of like a spring, it's then going to do what a spring does, right? So it's going to pop into its lengthened position, or in this case, it's going to pop toward the midline. And so that's the working or power strokes. That pulls actin toward the midline. Then, if we want to keep doing this, we have to attach an ATP molecule, and so that'll allow myosin to come off of those active sites on actin. So that's cross-bridge detachment. Again, cross-bridge is the interaction of actin and myosin. So to break those two apart, we have to add an ATP. And then we're going to break down the ATP, give off energy, and the energy that we give off when that's done allows us to reset the myosin head so that as long as those active sites on actin are exposed, myosin will keep interacting with actin. Okay? And so those sites will continue to be exposed as long as there's continuous calcium continuing to be released from the sarcoplasmic reticulum. Again, here's an embedded video if you want to check that out. All right, so before I move on too much from that, that is one of the things I really do like to ask about is the process of muscle contraction. So again, try to think about it, and I'll back up a little bit. Try to think about it in 2 phases. Back to this original drawing. So what happens at the neuromuscular junction? So again, the release of acetylcholine from the alpha motor neuron. Acetylcholine binds, causes the opening of chemically gated channels, get sodium come rushing inside. That causes the opening of voltage gated channels. Sodium comes rushing inside, and then we get that depolarization of the sarcolemma, spreads down the T tubules, spreads across the sarcoplasmic reticulum. That causes the release of calcium. Okay, and then that's kind of, so I break it up a little bit differently than this because so that's where I stop the first part. And then the second part is calcium binds to troponin C, pulls tropomyosin off of the binding sites on actin. Myosin will jump up and attach to those, pull actin toward the midline, we'll reattach an ATP, we'll break that down. That allows us to reset the myosin head. So be aware of those events and the order in which they occur. And I know that's super complicated, and I just did that in like 20 minutes. But that is something that takes some time for you to work with. So start drawing that out. One of the things that I had to do is kind of play with these diagrams the first time I was learning it, and kind of just go through the order of what all happens and when. And that was really helpful for me in terms of learning those things. And so obviously there's some pretty good diagrams in the book. Like, so this is figure 9.1. It's a pretty good diagram. And then I don't have the figure number here, but it's somewhere probably around 9.3, something like that. And then this is another important figure in the textbook as well. All right, so let's talk about muscle fiber types. So you can see there that broadly speaking, there are two fiber types. There's slow twitch, which are also referred to as your type 1 fibers, and then there are fast twitch, which are your type 2 fibers. Depending on the textbooks you look at, you'll see lots more types than that. We're just going to use the model that has 3 different types. So essentially you got slow twitch and fast twitch, and then under fast twitch you have 2 subtypes. So you have the 2As and the 2Xs. So one of the ways to think about slow twitch fibers are, these are the fibers that you use for endurance types of activities. So if you go out and you walk or you engage in a slow jog, those are your type 1 fibers that are contracting, that are propelling you forward. So slow twitch fibers are also plentiful in your postural muscles. So the muscles that hold up your head, you got a lot of slow twitch fibers in there. You also have a lot of slow twitch fibers in one of your calf muscles, the soleus. It's the deeper of the 2 calf muscles that are readily evident. So because that's the muscle, so when you're just, like I'm giving this lecture standing, when you're just standing there, soleus is the primary muscle at the ankle that's keeping you from falling over. And so it needs to have a lot of endurance. It needs to be fatigue resistant. And so it is primarily type 1 or slow twitch fibers. So looking at the muscle cells a little more closely, slow twitch fibers are obviously almost identical to your fast twitch fibers, but there are some important differences between them. One of those is that slow twitch fibers are smaller in diameter, so they're thinner cells. And so if they're thinner, they have fewer actins and myosins, and so they just don't generate as much force. So that's an important thing about them. So they're thinner cells, or they have a less diameter. So in addition to that, they also have lots of mitochondria. So remember, the mitochondria are the powerhouse of the cell. That's where we use oxygen to make ATP aerobically, and then we give off the byproducts of water and carbon dioxide. So your slow twitch fibers then are really good at using oxygen to remake ATP. They're also, so their sarcoplasmic reticulum is a little bit different than we see in the fast twitch fibers. Essentially, it is a little bit slower to release its calcium and a little slower to take it back up. And so the result is that slow twitch fibers take longer to generate maximum tension. So when we get that electrical impulse from the alpha motor neuron, release of acetylcholine, and all the stuff we just talked about, if we just get like one pulse that comes down let's go with a few pulses that come down the alpha motor neuron, cause depolarization in the muscle cell, and then we stop that stimulus, you're just going to get a quick release of calcium, quick interaction of actin and myosin, and then once we stop stimulating the muscle cell, we're going to pump calcium back inside of the sarcoplasmic reticulum, and so then we can't get an interaction of actin and myosin anymore. So that's a twitch. That's just one really quick contraction of the muscle cell. So slow twitch means that it takes a relatively long time for your type 1 fibers, or these types of fibers, to do that. So in terms of this diagram over here, what I want to call your attention to, so all the way over here on the right, these are your little slow twitch fibers. And so you'll see that in terms of the force that they can produce, so the highest number here is 10 grams. So at 100 milliseconds, they produce about 5 grams of force. So not very much force relative to, so this would be a type 2X fiber over here, all the way on the left. And so you can see that it generates 50 grams of force, and it does that in a little bit less than 50 milliseconds. So the type 2X fiber generates 10 times as much force as a type 1 fiber, and it does it in less than half the time. So that's where the twitch comes in. So this one is going to generate, the type 2X is going to generate maximal force really, really quickly, again in less than half the time as your type 1 or your slow twitch fibers. So, but that's not to say, give slow twitch fibers an entirely bad name, because they are super important. They're actually the fibers that you use for most things that you do every day. So your slow twitch fibers, the good things about them is because they produce their energy aerobically, they can produce a little bit of force for a long time, for hours on end. So they are highly fatigue resistant. It takes a lot to wear out your type 1 fibers. So that's the good thing. They're not very strong, but they can produce a little bit of force for hours and hours on end. So they got a lot of endurance, but they're not very strong. All right, so then let's go to the other extreme. Let's go to our 2Xs. So the 2Xs are the biggest and the fastest of the muscle fibers. So they've got a really big cross-sectional area, and that may be 30 to 40% larger than your slow twitch fibers. So pretty significantly larger than your slow twitch fibers. And again, in the case of a muscle cell, if you've got a larger diameter, most of that diameter is taken up by actin and myosin. So you've got more contractile proteins, and so because of that, that muscle cell can generate quite a bit more force. So that's why your type 2Xs, they got a lot more actins and myosins, they can generate your 50 grams of force. That's what makes them stronger. So other things that make them different from your type 1s, so they have few to no mitochondria, so they rely primarily on anaerobic energy production. So they're going to use the ATP phosphocreatine system, or they're going to use the glycolytic system, which is to break down sugar without oxygen. And so the good thing is that both of those, the ATP phosphocreatine system and the glycolytic system, are fast energy production. The bad thing is either you run out of fuel or you produce too much of a byproduct relatively quickly, and so the muscle cell wears out pretty quick. So for example, the ATP phosphocreatine system, it's remaking ATP using a molecule called phosphocreatine. Many of you are probably familiar with the creatine supplement. The idea is to have more of those creatine phosphates in the muscle cell so you can produce more ATP faster. So, but because both ATP and creatine are fairly large molecules, you can't store very much of them in the muscle cell, and so you would deplete your phosphocreatine store somewhere in the neighborhood of 10 seconds or less during like an all-out sprint. So type 2X fibers then run through their energy supplies pretty quick, so again, in only a few seconds. And then they can switch gears a little bit, and they can use the glycolytic system, so they can break down sugar without using oxygen, but a byproduct of that is lactate. And so then that's an acid, and so then the inside of the muscle cell gets acidic, and so that interferes then with the contractile machinery, that interferes with the interaction of actin and myosin, so you can only do that for so long too before the cell gets acidic and then you have to stop. So the type 2X fibers then can generate lots of force, but they can't generate it for very long. So they either run through all their fuel in the case of the ATP phosphocreatine system, or they produce too much lactate and then the inside of the cell gets acidic, and so then the cell can't contract anymore. So good thing, really strong fibers. Bad thing, not strong for very long. So few capillaries in your type 2Xs. Why that matters, so remember that your capillaries deliver blood to the cell, or more specifically, they deliver oxygen to the cell. So because your type 2Xs don't use a lot of oxygen, they don't really need very many capillaries. So few capillaries, few mitochondria in those, lots of glycogen, so sugar that they store inside of them, lots of ATP and phosphocreatine. And then again, their sarcoplasmic reticulum is a little bit different than what we see in the type 1s, where it's really quick to release calcium and really fast to pick it back up. And so that's what accounts for that difference in twitch speed. All right, and then the type 2As are kind of an intermediate between the two. So they're not quite as big and strong as the type 2Xs, but they're also not quite as fatigue resistant as the type 1s. So again, they're kind of a middle ground. They have more capillaries than the 2Xs and more mitochondria than the 2Xs, but not as many capillaries and not as many mitochondria as the type 1 fibers. So some other differences with them. So not only are the type 2Xs larger than what we see with the type 1s, but they also grow at nearly double the rate of your slow twitch fibers. So that's an important difference between them as well. So some other things, you'll see some shifting depending upon training. So this is somewhat malleable. depending upon the type of training that you do. So with, let's say, bodybuilding type training, where you do lots of sets of 12 and 15 on 5 exercises or more, what ends up happening then is you can shift the 2X fibers to act more like the 2As. So they will get more mitochondria, they'll get more capillary, but they'll lose some of their force production capacity. And some of that pertains to the nervous system's interaction with those fibers as well. Similarly, if you engage in that bodybuilding type training, your type 1 fibers will act more like the type 2As. So these fibers are going to increase in size, and they are going to get more glycogen, more ATP, more phosphocreatine, all of those things, and so they're going to be able to generate more force. If you engage in endurance training, so if you're a marathon runner or somebody who does lots and lots of endurance training, then everything kind of shifts this way and acts more like a type 1 fiber. So your fibers then, particularly the 2As, are going to lose diameter. They're going to shrink a little bit. And so why would that be advantageous if you're an endurance athlete to have smaller muscle fibers? Or why could it be? And one of the reasons that could be advantageous is because a muscle cell with smaller diameter, that's less distance for oxygen to diffuse into the cell, to get to the mitochondria, to make ATP, to fuel muscle contraction. So it makes it more efficient to produce energy aerobically the smaller the diameter of the cell. And you actually will get a little bit of shrinkage in the type 1 fibers with endurance training as well, or some studies have shown that. So again, malleable depending upon the type of training that you do to some extent. Now that said, most of us are give or take 50-50 in most of your muscles. And so your particular distribution depends upon how well you picked your parents. So elite endurance athletes will have more like 80% type 1 muscle fibers or 80% slow twitch muscle fibers in their quads, which is in part due to training, but also some of that is a genetic gift that they just had more slow twitch fibers than their counterparts did, and so then they just tend to do really well at endurance type sports. Conversely, your sprinters are kind of the other way, where they're going to have relatively few type 1 fibers, but they're going to have a whole lot of 2As and 2Xs. So the other thing about that is the more fast twitch fibers you have, again, because those can grow at double the rate and they have a higher upper limit on size. If you have a bunch more fast twitch fibers, you can get larger. Versus if you're somebody, if you lift weights, if you're somebody who has lots of, if you're that endurance athlete that has primarily type 1 fibers, well, they have a lower ceiling on their diameter. And so because of that, even with pretty extensive strength training, you won't be able to get quite as big as somebody who has a little bit different fiber type distribution than you do. So those obviously then play some role in affecting performance, but that's really becomes crucial when we're talking about elite performance. Like you're never going to win the Tour de France, which is an elite endurance event, if you have primarily fast twitch fibers. The other things are, as far as the fiber types go, I lost my train of thought. Oops. Let's see, what else? Pick your parents. What else was I going to say? I forgot. Doesn't matter. Apparently wasn't that important. All right. So anyway, that's what I was going to say. There it is. It depends upon the muscle that we're talking about. So different muscles will have slightly different distributions. So muscles that are more powerful muscles that we don't engage quite as often, so like pec major, will have a higher distribution of your fast twitch fibers. And so easier to make a muscle like that grow than, say, your soleus muscle, which again is an ankle muscle it's a postural muscle that holds you up. That is primarily type 1 fiber. So it also depends upon the muscle's job. Same kind of thing with some of the muscles in the neck and the back that stabilize your spine. Those are primarily slow twitch fibers. And so which we want them to have good endurance, but then there's not a lot of growth potential there. Not that any of us wants really gigantic spinal muscles. All right, so that's muscle fiber types. And then, so since I am about out of time, it looks like, yes I am, so we'll move on to motor units and motor unit recruitment on Wednesday. So we'll see you on Wednesday.